صباح الخير طلاب. Let us continue our talk about the metabolism of the glycogen, and we're going to start with the last point in the past lecture. I talked about the glycogenesis or glycogen synthesis of the glycogen. But before that, have a look for this figure 3. Structure of the glycogen has two chains, straight chain and branch. Straight chain and branch. Therefore, we need to form glycosidic bond 1,4 and glycosidic bond 1,6. Okay. Okay. Oh, sorry. Yeah. This is the structure. We need to form two types of the bonds, 1,4 and 1,6. Okay. The pathway of a glycogen biosynthesis or glycogenosynthesis or glycogenesis. We explained this point as in glycolysis. Glucose is phosphorylated to glucose 6-phosphate catalyzed by two types of enzyme, hexokinase in muscle and glucokinase in liver. Therefore, according to this step of reaction, glycogen synthesis starts after phosphorylated or phosphorylation step of a glucose to glucose 6-phosphate. But here we've got two enzymes. One of them, glucokinase, and the activity of the enzyme inside the liver. And the second enzyme, hexokinase, this is in another organism or outside the liver, the activity of this enzyme. Therefore, glucose is converted to glucose 6-phosphate in the presence of glucokinase and coenzyme magnesium +2 and 1 molecule of ATP to give glucose 6-phosphate. Okay? Kinase, that means addition phosphate group. Opposite side, glucose 6-phosphate will be converted to glucose by phosphatase. That means any phosphatase releasing for the phosphate group. Any kinase addition of the phosphate group. This is very important in any phosphorylation reaction. Okay, this is the first step inside the liver. Then now we've got glucose 6-phosphate. This glucose 6-phosphate unit is isomerized to glucose 1-phosphate by the enzyme. It's called phosphoglucomutase. Phosphoglucomutase. Now, glucose 6-phosphate in the presence of the phosphoglucomutase and the same coenzyme, the phosphate group is transferred from the carbon number 6 to carbon number 1. This is the second step in the glycogen synthesis. We need to phosphoglucomutase to transfer the isomerization, just transfer the phosphate group from the carbon number 6 to carbon number 1. So, glucose 1-phosphate reacts with a nucleotide, a certain nucleotide, which is uridine triphosphate, UTP, to form the active nucleotide, uridine diphosphate glucose, uridine diphosphate glucose, and pyrophosphate. According to this reaction, glucose 1-phosphate, all the reactions inside the liver, glucose 1-phosphate in the presence of uridine diphosphate glucose, pyrophosphate pyrophosphorylase, uh, and uridine triphosphate, UTB. The product is uridine diphosphate glucose, in addition to pyrophosphate. That means organic phosphate. Okay, this reaction it should be reversible, but because of one of its products, which is pyrophosphate, will be removed or will be hydrolyzed to two molecules of the inorganic phosphate in the presence of the inorganic pyrophosphatase enzyme. Therefore, one of the products has been removed or hydrolyzed. Therefore, the reaction undergoes to the product, to the uridine diphosphate glucose. So, the reaction is irreversible. This step, this information, very important in this step. Okay, now the result is uridine diphosphate glucose. Okay, and uridine diphosphate glucose, the structure of this compound, this one, uracil, nitrogenous base, and ribose is called uridine. Diphosphate, two molecules of phosphate. Glucose, this is glucose. The carbon number 1 in the glucose is active carbon. Okay, now here, we've got in this step, we've got an essential unit to start the addition of new molecules of the glucose to the primary glycogen using glycogen synthase enzyme, which is uridine diphosphate glucose. This is the essential unit, will react with the primer glycogen. Okay. Now, glycogen synthase enzyme catalyzes the formation of the glycoside bond between carbon number 1 of the activated glucose, as I said, of uridine diphosphate glucose, and carbon number 4 of the terminal glucose residue of the glycogen, liberating uridine diphosphate. Here, general reaction, we've got uridine diphosphate glucose react with the primer of the glycogen on the glycogen. The glycogen primer may in turn be formed on the protein primer, known as glycogen. And the glycogen is a 37 kilodalton protein that is glucosylated on the specific tyrosine residue 1,4 position to form short chain that is substrate for glycogen synthase. Okay, now here we've got uridine diphosphate react with the primer, glycogen primer is coming from the glycogen. In the presence of a glycogen synthase, the result is a new bond, 1,4 glucosyl unit, addition a new unit of a glucose, this one glucose, to primer, and the other result is uridine diphosphate. Have a look for this reaction. Now, we've got uridine diphosphate glucose, this one, uridine diphosphate glucose. The reaction in the presence of glycogen synthase between carbon number 1, this one, carbon number 1 of activated glucose, this one activated glucose, and carbon number 4 of the final residue of the protein in the glycogen. Okay? terminal glucose residue of the glycogen, liberating, releasing uridine diphosphate, liberating uridine diphosphate. And the result is primer, glycogen primer with the new units. This is in new units. Then this step will repeat with another molecule, uridine diphosphate. Then this carbon number will react with the final residue, and liberating or releasing uridine diphosphate. This step will repeat 6 times. This step will repeat 6 times until reach to length 11 glucose residue. Now the chains will be reached to 11 glucose residue. Then branching enzyme transfer. Have a look for this. When the branching enzyme, sorry, yes, after the chain will be reached to 11 glucose residue, like this one. 1 2 3 4 5 6 7 8 9 10 11. هذه new chain. After 11 glucose residue, branching enzyme will be active and transfer certain chain contains at least 6 glucose residue. This part or this chain will be broken by the branching enzyme. Then the branching enzyme will assist to bond this chain as a branch with the neighbor chain by the bond 1,6 linkage. Now breaking 1,4, forming 1,6. Okay, now each 11 new branch will be or certain chain consists at least 6 residue will be broken and 1,4 broken and form 1,6 as a branch. Therefore, here glycogen synthase added the glucose units. Now we've got a new chain, branching enzyme, a new 1,6. Have a look. This small chain consists of at least 6 will undergo as a branch to form 1,6 with the neighbor chain. Okay, inner chain have 2 alpha 1,6 branch. Okay, have a look for this figure. This is G glycogenin, which contains primer glycogen, and red color contains primer glycogen. Okay, inner chains have 2 alpha 1,6 branches. This is inner chains, 2 alpha 1 branches, 1 2. This is inner chains. Also this is for instance inner chains have 1 2. Also this one have 1 2 1 6 branches. Okay, chains in the outer tier السطح الخارجي are unbranched. Have a look. Unbranched. And they are 12 tier in mature glycogen particles. Now each mature particle of the glycogen must be consist of 12 tier. Here just appear just 5. 1 2 3 4 5. 1 2 3 4 5. 5, sorry. Okay, that means outer chain, outer tier are unbranched, and the mature glycogen particle consists of 12 tier. Okay. Now let us see the general diagram, for the general diagram for the pathway glycogen synthesis, glycogenesis, and glycogenolysis or hydrolysis of glycogen in the liver. Okay, we said glucose 6-phosphate will be transferred to glucose 1-phosphate. This is glucose 1-phosphate. Okay, in the presence of the uridine diphosphate glucose, phosphor or pyrophosphorylase and uridine triphosphate, the result is uridine diphosphate glucose, and the second result organic pyrophosphatase. And we said this reaction is irreversible because one of the reaction products will be removed or hydrolyzed to 2 molecules of the inorganic phosphate. Therefore the reaction is irreversible. Now this essential unit, uridine diphosphate glucose or nucleotide molecule, will react with the glycogen primer, which is on the glycogenin. Glycogenin is a protein, has a glycogen primer. This glycogenin will support the reaction with the primer of a glycogen. Then uridine diphosphate glucose react with the glycogen primer in the presence of glycogen synthase. The result is glucose. A new molecule of glucose will be added to the essential unit, 1,4. But the bond, a glycosidic bond, is 1,4 in addition to UDP. Then, after 11 units or residue of the glucose, the branching enzyme will be active to break certain chain has at least 6 glucose residue will be broken the glycosidic bond 1,4 to bond this new chain or this bonded broken chain to as a branch to the neighbor chain by the 1,6 glycosidic bond. 1,6 glycosidic bond. Here, and this step will be repeat glycogen synthesis at least 6 times until reach to 11, and the branching, each time there is a new branching. Each new chains consists of 11 glucose residue. There is a new branching. Okay, have a look for this half of the cycle is represented the glycogenesis or glycogen synthesis. Now we've got 3 hormones very important in the metabolism of the glycogen: insulin hormone and glucagon in addition to epinephrine. Now have a look for this compound, cyclic AMP. Cyclic AMP compound, adenosine monophosphate, it is very important on the enzyme glycogen synthase. I'm speaking about this side, just this side. Therefore, cyclic AMP is very important compound or a parameter affects on the glycogen synthase enzyme. This compound, it is always decrease the activity of the enzyme. Now, insulin will decrease the activity of the cyclic AMP or decrease the synthesis and activity of the cyclic AMP. Therefore, the activity of the glycogen synthase will be high. No parameter will affect negatively on this enzyme. Therefore, it will be normal or high. That means all the glucose unit will be transferred to glycogen. It is very normal because the insulin will be decreased. The function of the insulin will decrease the glucose unit or glucose level of the glucose in the blood. Therefore, all the glucose will be transferred to glycogen and will be stored in the glycogen. Okay. Opposite action, exactly, glucagon and epinephrine. Basically, these two hormones has positive effect on the cyclic AMP, increase the concentration and activity of the cyclic AMP. Therefore, this increase, that means will be decrease the activity of the enzyme. Increased cyclic AMP, that means the action of cyclic AMP will be more on the synthase, but negatively will decrease the glycogen synthase. Therefore, the glucose will be still in the bloodstream. No, not all the glucose will go to the form or to transfer to form glycogen. It is also very normal. Glucagon and epinephrine increase their function or increase the level of the glucose in the blood by affecting on the cyclic AMP positively, and this will decrease the activity of the enzyme. Therefore, not all the glucose unit will be transferred to the glycogen to store in the liver. It's very normal. In this side, I'm going to explain very short about this in this side, and you have to read more in the Harvard book. It's very clear and very short subject in the Harvard book. Now we've got glycogen in the liver. Glycogen in the liver. This side is represented this side represents the glycogenolysis or hydrolysis of the glycogen. Okay. Glycogen phosphate group, glycogen phosphorylase. This arrow represents many steps. Okay. But basically, using the two enzymes glycogen phosphorylase and glucan transferase, a deep branching enzyme, the result is a free glucose from deep branching enzyme. Okay, here one molecules will be released after deep branching of the enzyme. Finally, the result is glucose 1-phosphate. Okay, just one important points I want to explain it. Insulin and glucagon will affect on this enzyme, glycogen phosphorylase. Okay, insulin, its effect on the cyclic AMP negatively. Okay, therefore less amount of cyclic AMP concentration and activity. Therefore, activation of this compound will be less. Okay, activation of this enzyme will be less because cyclic AMP decreases the amount and activity of the insulin decreases the amount and activity of cyclic AMP. Therefore, this will be removed, and this enzyme will be, the activity of this enzyme will be decreased. Therefore, the glycogen, hydrolysis of the glycogen process will be decreased. It is very normal because insulin, its function remove the glucose, remove of the glucose from the bloodstream. Therefore, all the amount of the glycogen will be still as stored in the glycogen. That means insulin will not assist to release of the glucose from the stored glycogen in the liver. Okay, and opposite action for the glucagon and epinephrine. This is general procedure for glycogenesis and glycogenolysis, or synthesis of the glycogen and hydrolysis of the glycogen. Just very important, three hormones affect on the cyclic AMP, and we've got two enzymes are affected by the cyclic AMP. Here, very important point, this should be reversible, but the reaction is irreversible because one of the products will be removed by hydrolysis to two molecules of inorganic pyrophosphate. Glycogen is a complex protein and carbon. The final residue is tyrosine. Okay, give the glycogen primer to react with the UDP glucose. Okay, now let us see the types of glycogenesis disease. For example, or for instance, type 1, as I explained this type of disease, von Erich's disease, absolutely because of deficiency of the glucose 6-phosphatase, this enzyme, BGP6. Okay? And the symptoms, liver cell and renal tubular cells loaded with the glycogen. Hypoglycemia, lactic acidemia, ketosis, and hyperlipidemia, and I explained this why. For instance, for another disease, type 2, type 3, okay, for example, type 2, Pompe's disease, name of this disease, Pompe's disease, deficiency of lysosomal alpha-1,4 and 1,6 glucosidase because of deficiency in this enzyme, and fetal and circulatory accumulation of glycogen in lysosomes, heart failure. Even type 2 has a relationship with heart failure, maybe because of type 2. Also, type 4, amylopectinosis, Anderson's disease, its name, and the cause of disorders, absence of branching enzyme. We don't have a branching enzyme to form a branch or to break the glycosidic bond 1,4 and form 1,6 glycosidic bond to form a new branch. Okay, therefore accumulation of polysaccharide having a few branch points without branching, without the glycogen without branch. This is that means, as we said, the deposition of the glycogen in abnormal form, death due to cardiac or liver failure in first year of life. Here, in first year of the baby will be in dangerous case because of cardiac or liver failure. Yeah. Also, type 8 or type 7 Tourette disease, deficiency of phosphofructokinase in muscle and erythrocytes, and the characteristics is or symptoms as for type V النوع الخامس, but also possibility of the hemolytic anemia, يحصل عند anemia. Okay, this is very important types of disease, name of this disease, causes of this disease, and symptoms of this disease, and we've got type 1 to type 8. Therefore, the importance of the glycogen are very important in the biomedical field, because deposition deposition of the glycogen in the abnormal state will make a lot of disease, yes, as in this table we've got 8 types, but the causes are different sometimes because the process has a lot of enzymes. Therefore we've got a lot of types of disease, maybe because of deficiency in glucose 6-phosphate, or deficiency in the glycosidase 1416, or debranching enzyme, or branching enzyme, yeah, or deficiency of liver phosphorylase. Yeah, a lot of causes give a lot of types of disease have the relationship with the or glycogenosis. Okay, let us stop here this talk, and thank you for your listening, and don't hesitate. If you got any question, just send me, and absolutely I will answer you. Okay, brilliant. Bye bye.